Welcome students. Today we are going to throw light on peroxisomes and vacuoles. Now, what are peroxisomes? When improved methods of cell fractionation became available, in addition to lysosomes, a second group of particles called peroxisomes were isolated from liver cells. These particles are so called because they were characterized by their richness in peroxidase, catalase, D-amino acid oxidase and urate oxidase. Peroxisomes, though commonly found in liver and kidney of mammals, are also found in cells of protozoa, yeast and higher plants. In plants, following 4 types of peroxisomes are known. Number 1, glyoxysomes. Number 2, leaf type peroxisomes. Number 3, peroxisomes involved in urea metabolism. And number 4, unspecialized peroxisomes. Now, morphology of peroxisomes. Peroxisomes, also known as microbodies, are ovoid granules surrounded by a single membrane. Peroxisomes were discovered in 1966 by Didieu and his co-workers. They contain a fine granular substance condensing in the center to form an opaque homogeneous core, the nucleoid. The average diameter of a peroxisome in mammalian cell is 0.1 micrometer to 1.5 micrometer, and their number per cell may range from 70 to 100 as against 50 to 20 lysosomes per cell. They are particularly abundant in liver and kidney cells. Peroxisomes differ from mitochondria and chloroplasts in having a single membrane, and they do not contain DNA or ribosomes. Since peroxisomes differ from chloroplasts and mitochondria in not having their own genome and protein synthesis machinery, all their proteins must be imported by a process of selective import from cytosol. Although they are capable of self-replicating like endoplasmic reticulum. Now, chemical composition, biogenesis and assembly of peroxisomes. Chemical composition, biogenesis and assembly of peroxisome have been studied in detail in recent years. It is known that a number of proteins are associated with peroxisome, which help in a variety of functions that peroxisomes perform. Now, what are peroxisome membrane proteins and peroxisome proteins and protein receptors? There are more than 100 proteins that are either targeted to peroxisomal membrane or are transported across this membrane. Of these, large number constitute peroxisome membrane proteins (PMPs), and at least 32 known peroxisomal proteins called peroxins, which participate in the process of peroxisome assembly. Some of the genes that encode these peroxin include functions of peroxisomes. Since the peroxisome resembles mitochondrion in utilizing oxygen, it was earlier believed to be a vestige of an ancient organelle, which carried out oxygen metabolism in primitive cells having no mitochondria. It was also assumed that the development of mitochondria rendered peroxisomes less useful, since their functions were taken over by mitochondria. However, peroxisomes are now known to perform a variety of functions, including the following. Number 1. Removal of oxygen and production of hydrogen peroxide. Peroxisomes contain enzymes that use molecular oxygen to remove hydrogen from organic substances and produce hydrogen peroxide. This hydrogen peroxide is utilized by catalase to oxidize a variety of substances, including phenols, formic acid, formaldehyde, and alcohol. Catalase also converts excess hydrogen peroxide into water and oxygen. These oxidative reactions are particularly important in liver and kidney cells for detoxification of various toxic molecules that enter the bloodstream. For instance, 25% of ethanol we drink is oxidized to acetaldehyde in this manner. Number 2. Breakdown of fatty acids by beta oxidation. Peroxisomes achieve breakdown of fatty acids molecules by a process called beta oxidation. In this process, alkyl chains of fatty acids are removed sequentially by blocks of 2 carbon atoms at a time and are converted to acetyl coenzyme A. The acetyl coenzyme A produced during beta oxidation is exported from peroxisomes to the cytosol for reuse in biosynthetic pathways. In animals, this beta oxidation occurs both in mitochondria and peroxisomes, while in yeast and plant cells it occurs only in peroxisomes. Number 3. Photorespiration and glyoxylate cycle. In plants, 2 types of peroxisomes perform following 2 different but important functions. Number 1. In leaves, leaf type peroxisomes help in photorespiration, in which oxygen is used and carbon dioxide is liberated. Number 2. In germinating seeds, they convert fatty acids into sugars by a series of reactions known as glyoxylate cycle, so that these peroxisomes are called glyoxysomes. In this glyoxylate cycle, 2 molecules of acetyl coenzyme A produced due to breakdown of fatty acids are used to make succinic acid, which leaves peroxisomes and gets converted into glucose. This glyoxylate cycle does not occur in animals, so that they are incapable of converting fatty acids into sugars. Number 4. Enzymes and diseases caused by their deficiency. Peroxisome membrane contains 2 major proteins of 22 kD and 70 kD and several less abundant peroxisome specific proteins. The organelle matrix has more than 40 enzymes which catalyze a variety of reactions. These reactions can be anabolic, for example, plasmogen biosynthesis, bile acid biosynthesis, or catabolic, for example, beta oxidation of long chain fatty acids VLCFA. The hydrogen peroxide metabolism utilizes 3 enzymes: urate oxidase, D-amino oxidase, and hydroxylic acid oxidase in production of hydrogen peroxide, and a 4th enzyme, catalase, in its degradation. There are also interactions between intraperoxisomal reactions and extraperoxisomal pathways needing transport of metabolites across peroxisome membrane. Number 5. Peroxisomes and human disease. Peroxisomes are also responsible for certain diseases. These diseases can be broadly classified into 2 types: Number 1. Disease is due to deficiency of a specific matrix enzyme, which is called single enzyme deficiency. For example, ALD, adrenoleukodystrophy, and number 2, disease due to defects in the formation of organelle like Zellweger syndrome. In ALD disease, which is most common inherited peroxisomal disease at mid-childhood, there is onset of insufficiency of VLCFA coenzyme A synthetase. It has been shown that ALD is caused due to defect in a protein ALDP having a homology to a 70 kD peroxisomal protein PMP70 rather than to the expected VLCFA coenzyme A synthetase, and is involved in the transport of VLCFA coenzyme A synthetase across the peroxisomal membrane. ALDP belongs to ATP binding cassette ABC transporter protein superfamily. Now, evolutionary origin of peroxisomes. Evidences in favor of endosymbiotic origin of peroxisomes are actinobacterial origin. The reasons behind the actinobacterial origin of peroxisomes are: number 1, ability of peroxisomes to divide themselves and transport post-translational proteins. Number 2, presence of proteins in peroxisomes which are common to many prokaryotes. Number 3, peroxisome localized proteins PEX1 and PEX6 are more closely related to the cell division cycle proteins CDC48 homologs in actinobacteria than to the endoplasmic reticulum localized CDC48. CDC48 is a cell division cycle protein from yeast. Number 4, actinobacterial proteins show higher levels of similarity to those of the peroxisome than to those of other prokaryotes. Evidences against endosymbiotic origin and in favor of endoplasmic reticulum origin include the following. Number 1, peroxisome-less mutants can restore peroxisomes upon introduction of the wild-type gene through transformation. Number 2, homology between the peroxisomal import machinery and the ERAD pathway in the endoplasmic reticulum. Number 3, presence of a number of metabolic enzymes in the peroxisomes that were likely recruited from the mitochondria. Number 4, localization of peroxisomal proteins to the endoplasmic reticulum. And number 5, the similarity of some peroxisomal proteins to those localized in the endoplasmic reticulum. The evolutionary origin of peroxisomes still remains uncertain. Following two proposals are available. Number 1, symbiogenetic origin from a prokaryote, and number 2, endoplasmic reticulum origin as an imagination. Experimental evidence has been published for each of these two hypotheses. Prokaryotic symbiotic origin for peroxisomes was suggested on the basis of the following evidences. Number 1, the peroxisomal membrane is composed of purely eukaryotic bricks and is thus useful to trace the eukaryotic in their evolutionary pathway. And number 2, the peroxisomal matrix proteins import system shares mechanistic similarities with the endoplasmic reticulum and proteasome degradation process, indicating a common evolutionary history. Earlier, it was believed that The peroxisomes originate by budding from the endoplasmic reticulum and that its contents were imported from the cytosol. However, evidence is now available to suggest that new peroxisomes arise only from pre-existing peroxisomes by growth and fusion. Now, we will discuss vacuoles. Structure of vacuoles. The most conspicuous compartment in most plant cells is a very large fluid-filled vesicle called a vacuole. These are non-cytoplasmic areas present inside the cytoplasm and are separated from the latter by specific membranes. The term vacuole literally means empty space. It can be defined as a membrane bound organelle which is present in all plants and fungal cells. Vacuoles, although small and numerous, have been also found in animals and bacterial cells. Chemically, vacuoles are enclosed compartments which are filled with water containing inorganic and organic molecules. It is said that vacuoles are usually formed by the fusion of many membrane vesicles. Due to this reason, a vacuole does not have any specific size or shape. However, when it comes to the vacuole structure, it is designed so as to complement its function. Many mature and grown plant cells usually have a single large vacuole. There may be several vacuoles in a single cell, each separated from the cytoplasm by a single unit membrane called the tonoplast. The tonoplast is said to be a very active and dynamic membrane of this important part of plant cell structure. Generally, vacuoles occupy more than 30% of the cell volume, but this may vary from 5% to 90% depending on the cell type. The vacuole in its central hollow region contains a fluid known as cell sap. This cell sap contains different compounds, some of which are secretory and some are excretory in nature. Also, the vacuole in the middle, depending on the cell type and requirement, contains various concentrations of salts, sugars and different kinds of soluble pigments. This cell sap, which is a part of central vacuole structure, also contains various enzymes that are even capable of digesting the cell itself. These vacuoles arise initially in young dividing cells, probably by the progressive fusion of vesicles derived from the Golgi apparatus. They are structurally and functionally related to lysosomes in animal cells and may contain a wide range of hydrolytic enzymes. In addition, they usually contain sugars, salts, acids and nitrogenous compounds such as alkaloids and anthocyanin pigments. The pH of the plant vacuoles may be as high as 9 to 10 due to large quantity of alkaline substances or as low as 3 due to the accumulation of quantities of acids, for example, citric acid, oxalic acid and tartaric acids. Types of vacuoles. Depending on the contents and functions, vacuoles are of 4 types. Number 1, sap vacuoles. They are fluid-filled vacuoles or vesicles which are separated from the cytoplasm by a selectively permeable membrane called tonoplast. It has a number of transport systems for the passage of different substances. A number of small sap vacuoles occur in animal cells and young plant cells. In mature plant cells, the small vacuoles fuse to form a single large vacuole which occupies up to 90% of the volume of the cell. The large central vacuole spreads the cytoplasm in the form of a thin peripheral layer. This is a device for facilitate rapid exchange between cytoplasm and the surrounding environment. This fluid present in the sap vacuole is often called sap or vacuolar sap. It contains mineral salts, sugars, amino acids, esters, proteins, waste products and water-soluble pigments called anthocyanins and anthoxanthins. Some crystalline deposits may also occur. Number 2. Contractile vacuoles. They occur in some protestant and algal cells found mostly in fresh water. A contractile vacuole has a highly extensible and collapsible membrane. It is also connected to a few feeding canals, for example, paramecium. The feeding canals obtain water with or without waste products from the surrounding cytoplasm. They pour the same into the contractile vacuole. The vacuole swells up and the process is called diastole. The swollen contractile vacuole comes in contact with the plasma membrane and collapses. The collapsing is called systole. This throws the vacuolar contents to the outside. Contractile vacuoles take part in osmoregulation and excretion. Osmoregulation is required in freshwater habitats where water has tendency to enter the living cells. Due to the presence of higher osmotic concentration in the latter, continued entry of water shall cause bursting of the cells. This is prevented by throwing the extra water to the outside with the help of contractile vacuoles. Number 3. Food vacuoles. They occur in the cells of protozoan protestus, several lower animals and phagocytes of higher animals. A food vacuole is formed by fusion of phagosome and a lysosome. The food vacuole contains digestive enzymes with the help of which nutrients are digested. The digested material pass out into the surrounding cytoplasm. Number 4 is air vacuoles, also called pseudo vacuoles or gas vacuoles, and have been reported only in prokaryotes. An air vacuole is not a single entity, neither it is surrounded by common membrane. It consists of a number of smaller submicroscopic vesicles. Each vesicle is surrounded by a protein membrane and encloses metabolic gases. Air vacuoles not only store gases but provide buoyancy, mechanical strength and protection from harmful radiations. Now, we will discuss functions of vacuoles. Vacuoles play important metabolic roles. In addition to growth, these roles include the following. Storage. Vacuoles can serve as storage organelles for sugars, polysaccharides, organic acids and proteins. Most of the flavors of the fruits and vegetables are due to the compounds stored in the vacuoles. When needed, these primary metabolites can be retrieved from the vacuole and utilized in metabolic pathways. Toxic avoidance. Being immobile, plants cannot escape exposure to toxic elements in the environment by moving to another location. Nor do plants have an excretory system for the elimination of such substances. By accumulating heavy metals such as cadmium, the vacuoles can be weaved as a microkidney inside each plant cell, filtering and sequestrating potentially toxic ions from the cytosol. pH and ionic homeostasis. Reactions in the cytosol are delicately sensitive to changes in pH and ionic strength. The concentrations of certain ions, for example, calcium, are kept extremely low, enabling them to stimulate key regulatory enzymes such as protein kinases. The extremely low pH is due to the accumulation of sulfuric acid. In principle, the two proton pumps on the tonoplast can regulate cytosolic pH by pumping massive amounts of protons out of the cytosol into the lumen of the vacuole. Although this has been difficult to demonstrate directly. Presence of pigments. Many plant cells accumulate water-soluble flavonoids, pigments called anthocyanins, which range in color from orange-red to purple. In leaf tissue, such pigments are concentrated in the vacuoles of epidermal cells, where they probably function to prevent photooxidation of the photosynthetic apparatus by lowering the light intensity and by screening out ultraviolet radiations. In Petunia hybrida, flower petal vacuoles contain the pH-sensitive anthocyanin petunidin, which can exist in either a red or blue form, depending on whether the pH is acidic or alkaline. 4 complementary genes have been identified which have a bluing effect on flower color when they are homozygous recessive. It would be interesting to know whether the genes encode subunits of the vacuolar ATPase or PPase and whether they are specifically expressed in flowers. Dear students, this is all about structure and functions of peroxisomes and vacuoles. Thank you very much.